Hello everyone and welcome for another short video series. This is Ahmed Mustafa coming to you again. In today's presentation, I'm going to explain an important topic about the HLA antigen processing and presentation. And in this figure, I actually separated the class 1 from the class 2 into two different cells. But in a lot of cases, you can see that this can happen in one cell. So the reason I separated into two different figures just to make you sure or at least to know the difference on how does the class 1 and how does the class 2 antigen presentation happens or occurs. So let's talk first on the antigen presentation or antigen processing for class 1. So, for example, this is a cell that got infected by a virus, or could be a cell that undergo mutation by some sort of tumor. When this happens, the virus or the tumor antigen go through a proteasome degradation pathway. This will result in chopping up the virus or the tumor antigen into very small peptides, as you can see in this figure. This peptide will now enter into the endoplasmic reticulum through the TAP1 and the TAP2 channels. When it enters into the endoplasmic reticulum, this peptide will find the class 1 has a blank or an empty peptide binding groove, so automatically this peptide will bind to this peptide binding groove. And from the figure here, you can see that this is a clear class 1 because this is the alpha chain, and here is the beta 2 microglobulin, which I explained in one of my previous short videos. Once this happens, now the class 1 becomes fully mature. It will leave the cell and go to the cell surface, and where it's going to interact with a specific type of T cells called the CD8 positive T cells through the T cell receptor. Once this happens, the T cell become activated and it will come back in the form of a cytotoxic T cells and will kill the cells. And the reason why we want to kill these cells, because this cells is already been infected by a virus, or it could happen or a mutation happen in this surface, so we don't want the mutation to spread to other cells. So we need this is for the function of our immune system, that it comes and kill this infected cell. Let's now focus on the second part or the second cell here. Now you have an exogenous antigen. This is, for example, an antigen presenting cell. So this is a complete healthy cell. And the function of this antigen presenting cell is to capture or to sample blood, the surrounding, and take up any foreign organism. So, for example, if this cell took up this exogenous antigen, the exogenous antigen will enter the cell in the form of endosome, and then this endosome will fuse with the lysosome, and as a result of the lysosome and the enzymatic in the lysosome, this will decrease the pH of the endosome, and the decrease of the pH of the endosome will chop up or break down this exogenous peptide into small molecules. Let's go back to the endoplasmic reticulum to see what's happening here. Now, in this endoplasmic reticulum, you can see that the class 2, because now it's formed of an alpha chain and a beta chain, but you can see that the class 2 here is the peptide binding groove is not empty. It is actually occupied by another molecule called the invariant chain. And the reason of this invariant chain is to prevent these peptides from binding to the class 2. Remember, I told you that both class 1 and class 2 are in the same endoplasmic reticulum. So, and that's why if any peptides enter the endoplasmic reticulum, they only have one option to bind, which is the class 1, because the class 2 is already occupied by the invariant chain. And that's why we don't want peptides from the class 1 to bind to the groove of the class 2. So once this happens, the class 2 will leave the endoplasmic reticulum and will go to another compartment called the major histocompatibility class 2 compartment, where the invariant chains start to break and leave a small peptide or small molecule binding to the groove called the clip. Now this clip this HLA with the clip will go to another compartment where it meets another molecule called the HLA-DM. And here you can see that the endosome, which is full of peptide, will now come and fuse with this compartment. And now the function of the HLA-DM will come and remove the clip and allow the class 2 become blank. So now these peptides will find a spot to bind to, and it will bind to the class 2. And once this bind to the class 2 become mature, and it will go to the cell surface and activate another molecule called the CD4 positive T cells. This CD4 positive T cell, when become activated, it will be called the T cell helper cells. And the function of the T cell helper cell is to help other cells to function. So for example, here, this is an exogenous antigen, so we don't need to kill this cell. So when this cell become activated, it will be a helper cell. It will help the plasma cell to produce antibodies. These antibodies will circulate into the circulation and come back and kill or get rid of this exogenous antigen. So what is the clinical significance of these two pathways? So for example, if this pathway become activated after transplant, this will increase or will result in what we call T cell mediated rejection. If this pathway become activated after transplant, this is what we call as antibody mediated rejection. And in some cases, you can find that two pathways are both activated, and this will result in what we call mixed T cell mediated rejection and antibody mediated rejection. Thank you again for another short video.